Today I'm going to try and tell you a little bit about adult Refsum disease, its clinical features, diagnosis and treatment. Adult Refsum disease was first recognised about 50 years ago as a distinctive clinical condition comprising a number of features. These involved defects in the eyes, the hearing and the sense of smell, and were associated with feelings of weakness and altered nerve sensation. So this gives rise to a cluster of syndromes, or features, or a syndrome. And the syndrome can have many causes, but it can also have a single one. And originally Refsum disease was thought to have a single cause. So the clinical features of Refsum disease are night blindness, where you lose vision, particularly at night, and are able to distinguish and start bumping into things. That is associated with changes at the back of the eye, called retinitis pigmentosa. There is also a loss of sense of smell and taste, though people may not actually notice this until this is formally tested. When the disease is severe, you can have defects in nerve sensation and in nerve function, so that you are able to touch and feel things properly, or get weak, and those weakness can extend in severe cases to problems in the heart with heart rhythm changes. You can get changes in the skin with the development of a large scaly rash called ichthyosis, and also in some people there are seen to be bone defects which are present from birth, particularly short fingers and toes. And these have all led to a battery of diagnostic tests that we apply in the diagnosis of Refsum disease. So in the case of vision, we look for cataracts, we look for whether pupils react, we look at the extent of any retained vision and how wide the visual fields are. We test smell with a panel of smell tests, either a number of bottles or these days a scratch and sniff test card called the upsit panel. We test nerve function, so can people feel different types of sensations, and how fast do nerves transmit when we record them. That's a neurophysiology test. Similarly, we can test muscle function. We can do tests in the blood, and the one that is key for Refsum disease is measuring a complex fatty acid with branched structure called Phytanic acid. And lastly, and most recently, we can do gene tests to look for defects directly if we see any of these features, as it might be caused by Refsum disease. So the features have a varying onset between individuals and with age. So retinitis pigmentosa, loss of vision, is commonest. It typically becomes obvious around age 10, but rapidly progresses so that virtually everybody has severe visual loss by age 40. And this graph uses diagnosis at age of blindness as the criterion. So similarly with loss of smell, we test it, it's there in almost everybody, but other features such as loss of nerve sensation, deafness, and skin changes are so. And similarly, when you actually have a loss of sensation in some of the nerves that control balance, you actually have ataxia and start staggering, and that is just another form of neuropathy. And again, as you can see, that is only present in about half the patients, and these are all those who came in acutely ill originally. Now in the eye, these are the kind of things that we would see through an ophthalmoscope, or these days a camera. So instead of seeing a nice pink background with nice red blood vessels and a yellow fovea where the nerves come in, we see this splotchy pattern of black pigment coming through from the back of the eye, and that is a retinitis pigmentosa. Typically in Refsum disease it's called a salt and pepper distribution, with this sort of alternating spots of white and black. And it starts peripherally and gradually comes in. When we look at the extent to which people can see, not only in terms of their peripheral vision, you gradually in Refsum disease see tunnelling in of the vision, such that in the field tests you see that all that remains in this particular individual is actually the fovea, the bit that actually sees colour, as all that is preserved, with almost total loss of visual sensation outside this. The bone features, if they're present, are highly characteristic, and these are short fingers and toes, particularly short thumbs, on fifth or fourth toes. And these can be clearly seen in this individual through photographs, but sometimes we have to do x-rays to see them because the changes can be a little bit subtle. So the traditional method of diagnosing Refsum disease has been to look for the clinical features, so the retinitis, the anosmia, the neurological finds and the bone changes. And that used to be how we found most of the cases. And we looked for those, particularly in people that are admitted acutely unwell with symptoms of weakness. And that's been a presentation of most of the cases of Refsum disease that we saw up till about maybe 15 years ago. When we only test their bloods, we find that they have very high levels of phytanic acid, and we can also do more detailed studies after taking a skin biopsy, growing the cells, and looking at the function of the enzymes that degrade phytanic acid and showing they're absent, and those are called fibroblast enzyme studies. However, things have moved on, particularly in the last 5 years or so. Genetic diagnosis has become quick, it's become easy, and it's become something virtually any specialist can order, and in some countries, primary care is now beginning to order these tests. And now we have panels of genes that are associated with various clinical features, and the retinitis pigmentosa panels, for instance, commonly contain a test that looks for PhyH, the defective enzyme in Refsum disease. Similarly, some deafness panels, but those are not exclusively, also contain PhyH, as it can be a cause of later onset deafness. And we do these tests first and then go on to formally confirm that the test is correct, that the change we found is actually pathogenic, a mutation and causative, and by measuring the blood phytanic acid levels, which are severely elevated. Now, there are other things that can look like Refsum disease. As I said, it's a syndrome, it's a cluster of features, and clusters of features can have multiple causes. And we know that there are other causes that cause the particular cluster called Refsum disease. And those can be either defects further down in the pathway, enzymes that are a bit further down, and they give rise to different changes. So maybe not the phytanic acid is raised, but something called Phytanic acid, which is about 2 steps further down. There are some that we know we can make as mouse models, but we've so far not yet found in any humans. So those are still people that are probably waiting to be discovered. And then we have a whole cluster of things that cause the same clinical features, but don't seem to have anything to do directly with Refsum disease. So we have the Usher syndromes, which are normally known as being related to congenital deafness, and those can be associated with Usher 2B in certain times with retinitis pigmentosa. We can have defects of aldehyde metabolism, such as Stogen-Larsen syndrome, and we can have defects of endocannabinoids metabolism, such as PhyH, that have been described in individuals. And all of these can give rise to syndromes that share many of the clinical aspects of Refsum disease, even to the extent of having a slight elevation of phytanic acid, which we see in Stogen-Larsen syndrome, but we don't necessarily see in Usher, or PhyH. So what is it that causes Refsum disease? Why do we run into this problem? Well, Refsum disease is a defect in the ability to metabolize the products of chlorophyll. So green leaves contain chlorophyll, which converts light into sugars. And the problem comes when animals eat plants which contain chlorophyll, or fish eat algae that contain chlorophyll. And because those animals metabolize the chlorophyll, then they also break it down into its components. It comprises a light sensor ring, a set of 4 rings, and an anchor chain. And the problem with Refsum disease is all to do with the anchor chain. So in this graphic, you can see the rings in the top right, which are the chlorophyll, and this little anchor at the end with the spikes of methyl groups every 4 carbon units. And that gets cleaved off from the chlorophyll, separated from the chlorophyll through bacterial degradation, as occurs in the animal or fish gut. And that is made into an alcohol, that releases an alcohol. And just like you make wine into vinegar, you can oxidize up alcohols to acids, and that the bacteria also do. and they make it into phytanic acid, and phytanic acid is what is toxic in Refsum disease. Now, phytanic acid has this branch structure of these regular spikes of methyl groups every 4 carbons, and it has an acid, a double OOH, at the termed alpha end. And generally, most fatty acids are broken down from the alpha end, and they're broken down by removing 2 carbon units. Unfortunately, if you attempt to remove 2 carbons from the alpha end here, you see you run into a block, which is highlighted. So therefore, that pathway is blocked. So how do animals deal with this? Well, they actually remove the OOH, the first carbon, and thereafter these cutting enzymes skip the methyls and can degrade the fatty acid in 2 carbon units as they normally would. And that is the block in Refsum disease. This first alpha oxidation, this oxidation of this first carbon to actually remove the acid group is the problem. Alternatively, there is another pathway that starts at the other end, but what that does is it creates an acid group at the other end, and because the block is now in the wrong place, it is just a little bit closer to the carbonyl group, the 2 carbon cutting unit works perfectly, so you can degrade normally, and that is called omega oxidation. So in Refsum disease, it is a recessive disease. That means you actually have to have 2 defective genes for the symptoms to express, and therefore it is rare. It's about 1 in a million. 90% of the defects are caused by a defect in the enzyme that drives alpha oxidation, the phytanyl-CoA hydroxylase, or PhyH for short. About 5% of the defects are driven by an inability to put the enzyme in the right place. Fatty acids are degraded in a special compartment within the cell, which is actually full of oxidizing agents, which you don't want to get around everywhere. And that special compartment is called the peroxisome. And if you cannot import the enzyme into the peroxisome, and it does not work, and therefore you'll get the symptoms. And then we have a number of other conditions that may or may not cause phytanic acid elevation, which cause the syndrome, as I previously mentioned. The defect is in the enzyme, which we have both identified, we've managed to crystallize, and we've managed to actually find out what the structure looks like. And most of the defects, about a third of them, are actually due to splice defects. Genes in man and in higher animals are encoded in DNA. They're encoded in short chunks of coding sequence, which are then spliced together to make a final transcript. That final transcript is used as the basis of making the protein. If you can't make the splice sites, or the splice sites don't work, then you can't make the enzyme, or it becomes garbled, and therefore it won't work. And about a third of the defects are of that case. About another third are caused by defects in the enzyme itself. The enzyme has 2 sites. It's got a binding site for phytanic acid, which actually exists inside a carrier protein. It also has a site for binding something that comes from sugar metabolism called 2-ketoglutarate or alpha-ketoglutarate, and that binds to an iron atom. You put the 2 in too close proximity, and that enables the excess carbon to be transferred, in actual fact broken and released, to actually release molecular oxygen. And that is the 2-ketoglutarate binding site, and many of the defects in Refsum disease are actually in the binding site, which actually physically distort the structure, so ketoglutarate won't bind. And that's about a third of the defects. The other third we really don't, we can't characterize, but they probably involve subtle effects around the process. As I previously mentioned, you can also have Refsum disease caused by an inability to put PhyH in the right place. So it can't go into the peroxisome, this oxidizing environment where complex fatty acids are broken down. And PhyH binds to a carrier protein called PEX7, and PEX7 is transported through a complex piece of machinery into the peroxisome. And therefore, if you have a defective PEX7 binding site, either in PEX7 or in PhyH, you can't enter the cell, and therefore it won't work, and you get Refsum disease. So, given that we know that Refsum disease is caused by a fatty acid that comes in from the diet, phytanic acid, and it's not made in very large quantities in man, to our knowledge, then how can you treat it? Well, you can remove it. Fatty acids dissolve in fat, but there are fat particles in the blood, and we can remove those, and that's what we do with dialysis for fat or apheresis. We can also restrict intake, because this is all coming in from dairy products or herbivore meats or high-level fatty fish. We know that we can restrict that, and that will actually mean that the stores do not get topped up, and therefore you'll not get toxicity. Problem is that though most of the Phytanic acid in man gradually accumulates in fat tissue, we do maintain a store of it in the liver, because we maintain a store of acute liver fatty acids for energy purposes. That's what we use for rapid energy requirements for when, for instance, about 30 seconds after going for a run, where we switch over from sugar metabolism in the form of glycogen to fatty acid metabolism. And one of the things that we can do is, as you start burning that fat from the liver, is that's released acutely and increased to stress. And therefore we have to manage that metabolic stress. Because if we can damp the liver pathways and prevent a spike of Phytanic acid being released, and it's the spikes that actually contribute to the toxicity, or release that contribute to toxicity, then we can manage the symptoms. And we do that by either giving sugars to suppress fat metabolism, that's dextrose infusions, or we give proteins, because proteins can be turned into carbohydrates and sugars and for the same purpose. And chronically, as I've mentioned, we take out things that contain fatty acids, and Phytanic acid in particular, and that's herbivore meats and dairy products. And when we do this, we can observe a fall in Phytanic acid levels. This is a graph showing a patient over the period of admission of about 20 days, and then their follow-up up to about 40 days after admission. This is a patient who came in acutely unwell, had developed weakness, was unable to stand, was staggering, had lost sensation in their hands, and had already been noted to have visual signs of retinitis, but no one had made the diagnosis. And you can see their original Phytanic acid levels are about 2,500 micromoles per litre, which is about 100 times what they ought to be, which is about 25. And you can see in this case, this was done with diet alone, just restricting, and we did a couple of rounds of plasmapheresis on the day 3, 2 and day 6 in this case. We got a level of overall fall in Phytanic acid levels down to about 20% of original levels by 40 days. Now this is accomplished by a mixture of restriction, giving calories to stop Phytanic acid being broken down while they were acutely ill, but also the patient's own response through activating this bypass pathway called omega oxidation, which does exist in humans to degrade fatty acids. And it's not specific for Phytanic acid, it will deal with any fatty acid which is present in excess. And here omega oxidation was very active to begin with, and then rapidly falls off in activity. But the interesting thing here is that it was induced early. And then the question arises, can we induce this with a drug? Because in that case, we might have a potential treatment. We've been trying to do this for a number of years, and it still remains an active amount of research. I mentioned that we can take fatty acids out through dialysis, and this is the kind of typical spike and drop picture that you see with dialysis. So this patient received 2 bouts of dialysis with their Phytanic acid going up markedly prior to dialysis, then dropping with the dialysis, and then coming up again as you got Phytanic acid release from the liver and from the fat acid stores. And then the longer term, they're managed with diet. So the principles of a diet in Refsum disease are well established. You avoid rapid weight loss, you eat regular meals, and you gain weight if you're underweight. All of this stops you breaking down and releasing the liver's acute store. You avoid diets rich in Phytanic acid, you eat plenty of fruit and vegetables, and you don't take caffeine drinks in excess, because caffeine and caffeine-related products, such as found in some of the energy drinks that are supplied these days, can cause acute spikes of Phytanic acid. Nutritional supplements that can give rise to carbohydrate are used in emergencies when people are ill or when they're due to starve for surgery or other purposes. And lastly, we offer specialist advice, because specialist can actually help keep people out of hospital and advise on the details of diets. And if you follow a long-term phytanic acid diet, this is over a period of about 20 years, you can see that gradually the phytanic acid levels fall from about 100%. Most of these patients start level at about 1,000 to 2,000, and they gradually fell with time with these spikes. And these spikes are generally intercurrent illnesses or surgical admissions for causes unrelated to Refsum disease, but each of which caused a spike in phytanic acid. And as they went on the diet for longer, the spikes became less prevalent, they also became smaller, so therefore people actually responded better to what we call metabolic insults, whether those are infections or surgery or any other cause. So actually the diet is very helpful in the long term. Now we know that fasting is bad for patients with Refsum disease. We were fortunate enough to be able to conduct a study under controlled circumstances in a hospital bed. I wouldn't advise anything outside that. And where 5 people agreed to fast for 48 hours. Not a particularly long time, given that we now see some diets recommending 5 and 2 fasting regimes for weight loss at the moment. And what this did in people with Refsum disease was it doubled their phytanic acid every 30 hours. So this time, virtually all of them received levels 4 times what they had originally. We saw that the omega oxidation pathway was induced, again, as we saw in that acutely ill patient. But the interesting consequences are what happened afterwards, because we did this study under controlled circumstances. We actually kept patients in for 5 days. This was in a metabolic bed, so we were fairly certain that we wouldn't cause any acute problems because they were under observation and being checked. But then we actually also followed them out for about another 3 months. And there we saw different changes. We found patients spiking their phytanic acid, not without any symptoms we could see. Another had an episode of weakness. Whether this was associated with that previous episode of fasting or not, we don't know. It might have been an intercurrent infection. And one started losing weight and lost quite dramatically, lost weight for no obvious reason. And this is another reason why we discourage people from going on fasts with Refsum disease. Some of the consequences are not predictable and they may occur a while after the episode of prolonged fasting. So in conclusion, Refsum disease is a rare and easily identifiable condition. It's got a very diagnostic cluster of features: the retinitis, the anosmia, the bone changes, and the raised phytanic acid. It's got an established treatment, which is the diet, which will slow the rate of deterioration, and if we drop phytanic acid levels in the acutely ill, then things like the nerve defects, the muscle defects, the heart rhythm defects, and in a slower time course, the skin disorders will all resolve. We will see a slowing in the rate of deterioration of vision and a slowing in the rate of deterioration of hearing with time, as people who are on better diets and achieve lower levels seem to deteriorate slower than individuals who maintain high levels. Now, these are all treatments that are based on physical interventions, whether it's diet or plasmapheresis or a combination of the two. Ideally, we'd want some drugs that actually delivered some of the same benefits, like through inducing omega oxidation, but we haven't found those as yet. Or we could fix the defect ourselves exogenously, and that's where the idea of doing gene therapy comes in. And as much of the problems with Refsum disease come in from that defect of fat acid handling in the liver, and this store of phytanic acid that can be released acutely in response to stress and infection or illness or surgery. And if we could actually alter liver metabolism by putting the gene back in, maybe we could deal with many of the acute illness features of Refsum disease. It might even help the visual signs, where gene therapy is also possible in the eyes. So we have a condition here that is rare, it's easily identifiable, and it's got some reasonable degree of treatment. Thank you very much.